This is part 2 on megaloblastic anemias. Let us talk about what the laboratory actually shows us in megaloblastic anemia. One of the most important things to understand up front is that the laboratory findings in the peripheral blood and the bone marrow are essentially identical across all forms of megaloblastic anemia. It does not matter whether the cause is B12 deficiency or folate deficiency, the morphologic picture looks the same. This is why measuring serum vitamin levels is essential to make the distinction, because the microscope alone cannot tell you which vitamin is deficient. In the peripheral blood, 3 findings are classic and stand out immediately. The first is pancytopenia. This means all 3 major blood cell lines are reduced simultaneously. Red cells, white cells, and platelets are all low. The reason all 3 are affected is that B12 and folate deficiency impaired DNA synthesis throughout the bone marrow, not just in red cell precursors, so all rapidly dividing hematopoietic cells are affected. The second peripheral blood finding is oval macrocytosis. The red blood cells that do make it into circulation are abnormally large and oval-shaped. Quantitatively, the mean corpuscular volume, the MCV, which is the average size of a red cell, is often greater than 110 femtoliters. To put that in context, the normal MCV is approximately 87 femtoliters, so these cells are substantially oversized. This macrocytosis reflects the impaired cell division. Because the cells cannot divide properly, they keep growing without splitting, producing these oversized cells. The third and very distinctive peripheral blood finding is hypersegmented neutrophils. Normal neutrophils, the white blood cells that are part of our immune defense, have nuclei divided into roughly 3 to 4 lobes. In megaloblastic anemia, the neutrophils develop with more than 5 lobes in their nuclei. These are called hypersegmented neutrophils, and they are a classic highly specific marker of megaloblastic anemia. Spotting them on a blood smear is a strong signal that something is wrong with B12 or folate metabolism. In the bone marrow, the finding is megaloblastic hyperplasia, meaning the marrow is packed with those abnormal large precursor cells we discussed earlier. There is an overaccumulation of megaloblasts reflecting the ineffective production process. Now, to distinguish between B12 and folate deficiency specifically, serum levels of both vitamins must be measured directly. The morphologic picture in the blood and marrow is not able to make that distinction for you. The specific type of megaloblastic anemia is determined by which vitamin is found to be deficient on laboratory testing. In the next section, we will talk about pernicious anemia, the most common form of B12 deficiency.